All right. I think we can go ahead and get started. Hi, everyone. Welcome, and thank you for joining us for Vector Builder's Gene Delivery Seminar Series. Our speaker today is Dr. Mike Kalwat, Scientific Director at the Indiana Biosciences Research Institute. After receiving his bachelor's degree from Purdue University, Mike completed his PhD at the University of Indiana, where he studied cell responses to secretory stimuli in pancreatic beta cells. This was the start of a career studying beta cell function in diabetes and other diseases that's seen Mike through a postdoc at the University of Texas Southwestern Medical Center and the start of his own lab at the IBRI. There, his lab uses high throughput screens and a variety of cell, molecular, and omics approaches to study the regulation of hormone secretion. Following the presentation, we're going to have a Q&A session, so please type any questions that you have into the Q&A text box at any time during the presentation, and I'll read them out at the end. And now I'll hand things over to Mike.
Thanks, Connie, and thank you to Vector Builder for giving me the chance to present some of my work today. This might be a little change of pace to what you're normally accustomed to. While we do use Vector Builder in our work, what I'll talk about today does span a range of various molecular and cellular biology techniques. There we go. So I'll just give you a little outline of what I plan to go over today. So I want to talk a little bit about beta cell dysfunction in diabetes and in a rare disease called congenital hyperinsulinism. So I'll give you some background on that, and I'll tell you about a high throughput screen we did where we identified a small molecule that causes beta cells to hypersecrete insulin. And then I'll describe some approaches we took to identify the target of that compound and use it as a tool to study the phenotype of hypersecretion in beta cells. And at the end, I'll touch on a new project that I'm working on with one of my collaborators to apply machine learning to single-cell RNA-Seq data from human islets to try to understand beta cell heterogeneity, which impacts diabetes. So needless to say, diabetes is a worldwide health problem, not just in the United States, where over 11% of the population have diabetes, but across the world, over 500 million people have diabetes, and it's only projected to increase over the next 5 or 6 years. So in order to understand what goes wrong in diabetes, it's important to understand the beta cell because the beta cell is one of the key players in the development of the different forms of diabetes. So when you eat a meal, in this case, some ice cream bars from Costco, blood glucose increases, and the beta cells sense this increased blood glucose and release insulin in response. Insulin travels to the peripheral tissues like skeletal muscle and adipose tissue to promote glucose uptake, thereby regulating glucose homeostasis. However, in different forms of diabetes, the beta cells become dysfunctional. For example, in type 1 diabetes, there's an autoimmune attack that kills beta cells, leading to insulin dependence. This accounts for about 10% of the diabetes cases in the United States. But a majority of the cases are actually type 2 diabetes, which is caused by an intricate interplay between insulin resistance and beta cell dysfunction. They kind of go hand in hand. And over time, as an individual gains weight or becomes more insulin resistant, the beta cells are able to compensate for this by expanding their mass and producing more insulin. But at some point, in some individuals, the beta cells are no longer able to maintain this compensation and they fail. And once insulin production drops enough, you get hyperglycemia and all the downstream effects of diabetes, including kidney disease, blindness, nerve damage in the peripheral tissue, and cardiovascular disease. So our goals in my lab are broadly to understand the pathways that regulate beta cell function and develop molecules that could lead to improved beta cell health. And finally, to better understand the heterogeneity of beta cells within individuals and across individuals. And so along those lines, we want to know what what are the intrinsic factors that could contribute to beta cell failure in diabetes. And I mentioned insulin hypersecretion, and I want to mention that it's a feature of type 2 diabetes as well as rare diseases, which we'll talk about next. Before developing type 2 diabetes, you can have a raised insulin secretion called basal hyperinsulinemia. And when this occurs, it actually increases the risk of developing type 2 diabetes. And there's also multiple published evidence supporting the fact that insulin hypersecretion could precede beta cell failure and probably drive disease pathogenesis. For example, there are studies of using sulfonylureas, which is our anti-diabetic drug that makes the beta cells secrete more insulin. Chronic treatment with these drugs can promote beta cell failure in the long term. And there are also genetic studies showing that humans with mutations in certain genes can exhibit hypersecretion prior to beta cell failure. And I mentioned the rare disease congenital hyperinsulinism in my outline. The reason this disease is interesting to study is because it's actually like the opposite of diabetes. So in these patients, there are mutations, genetic mutations that affect beta cell function such that they chronically secrete insulin when they're not supposed to. This affects about 1 in 25,000 births, and there are very few treatment options available for these patients. Usually they end up having to have large parts of their pancreas removed to treat the disease. So studying this rare disease not only helps us understand how beta cells work in general, it might also let us develop therapies for these patients. So overall, my lab is focused on beta cell function, and we take chemical approaches, and we use beta cell lines as well as primary islets from human donors. And we study these pathways that cause insulin secretion and that lead to various stress responses in the beta cell. And I'll just briefly mention a little bit about how beta cells sense glucose and secrete insulin. Glucose in the blood enters through a glucose transporter on the cell surface. And after metabolism through glycolysis and the TCA cycle, elevated levels of ATP can cause a special potassium channel in the membrane called the KATP channel to close. That leads to membrane depolarization and the opening of voltage-dependent calcium channels. This calcium influx is what triggers insulin to be released. There are also other metabolic intermediates that are formed that amplify the cell's response to this calcium influx. So we use different chemical probes to drive this process and sometimes put it into overdrive so that we can see how the cells respond by inducing either adaptation and recovery, or they can, if there's too much stress, it can lead to cell death. And we also integrate various omic approaches like transcriptomics to look at gene expression over time, and publicly available omics databases that are contributed by various labs across the country. So I'd like to move on to telling you about how we identified a small molecule that causes insulin hypersecretion. So when I was in my postdoc, I wanted to find a way to study insulin secretion cheaply and quickly without having to pay so much money for ELISAs and take a long time to see the results. So we developed an assay where we took advantage of this approach. It was actually published by researchers at the Broad, where you express in beta cells a insulin secretion reporter, and that reporter is actually the protein insulin with a secreted luciferase inserted into the C-peptide. And when insulin is normally processed, this C-peptide is excised in the granule. So you end up with a full-length insulin that's formed by these A chain and B chain, and then you end up with this Gaussian luciferase floating around in the insulin granule. These are co-secreted when the cells are stimulated with glucose, and you can take the culture supernatant and run a very cheap and easy luciferase assay. So we made a stable beta cell line that expresses this reporter, and we confirmed that it responds to glucose normally, and that the secretion of this luciferase is well correlated with endogenous insulin secretion. And we took this reporter cell line and we took advantage of the fact that we had over 100,000 compound library at UT Southwestern. And the design of my screen was that we exposed cells for 24 hours to compounds in a search for compounds that chronically modulated beta cell function. And so after that 24-hour exposure, the compounds were washed off and cells were starved and stimulated before we did our luciferase assay. And we ended up finding about 150 compounds that affected beta cell function. And we spent a long time in the core using these liquid handlers, doing lots and lots of these 384 well plates. And I had a lot of help from my research associate, Dirk Binns. So most of these compounds turned out to look like inhibitors. And so at first we thought that we mostly found inhibitors in our screen. But when we went and counter-screened the top inhibitor hits and we exposed cells only for 1 hour to the compounds, we found that actually a subset of them potentiated insulin secretion, enhancing it. And that this probably is related to why they become inhibitory after a longer-term treatment. So we took one of these top candidates that had this phenotype called SW16789. And once I started my lab, I had these two talented researchers, Geetan Jolly-Roy and Karina Rodriguez dos Santos, help me work on this project. So after we had identified this compound, we needed to characterize it a little bit better to figure out what was going on. And we found that if you treat the cells for 1 hour or 2 hours with this compound, it can potentiate the amount of insulin that's released in response to glucose. But after only 4 hours, actually, or longer, up to 24 hours, the cells will no longer respond. So they basically have this refractory response after experiencing this hypersecretion. And I told you earlier about how the beta cell secretes insulin. It uses calcium influx to release the insulin. And so we can measure calcium influx using a Fira2 dye loaded into the cells. And if you treat the cells with glucose and the drug acutely, you can see that the SW16789 is able to amplify the amount of calcium influx in response to glucose. And if cells were treated overnight first, and then the compound was washed off, the calcium influx is partially suppressed. But it's not gone all the way. So this calcium can still come in, but the cells aren't able to respond by secreting insulin. And we also confirmed that these findings were consistent in human islets. If we exposed human islets overnight to this hypersecretion-inducing compound, the following day after washing off the drug, the islets no longer responded to glucose. So we tested a lot of different things, and I don't have time to go over all of them today. So I'll cut to the chase. Our search for the mechanisms of action of this compound led us to calcium influx. And so how do we narrow in on how this probably is the key mechanism of action for this compound? We did a chemical suppressor screen. And so what we did was if we treat the cells overnight with either a vehicle or the hypersecretion-inducing compound, and we also co-treated with a variety of other different inhibitors, different pathways to identify which drugs could protect the cells against this hypersecretion stress. And one of the only classes of drugs that was able to protect the cells against this stress were voltage-dependent calcium channel inhibitors. And this one I'm showing you is Nifedipine. And so if you give the cells the hypersecretion compound overnight, you can see they lost their ability to secrete insulin. Where if you co-treat with Nifedipine, you're able to protect some of that response. And we found that this was also consistent in human islets under the same conditions, suggesting the pathway is conserved in mouse and human beta cells. And interestingly, this isn't the only compound that can cause this hypersecretion stress. So I don't think this is unique to the compounds we found. It's a conserved pathway in beta cells. So there are other drugs, like I mentioned sulfonylureas earlier, that inhibit that potassium channel and cause more insulin secretion. There are also derivatives called like dextrorphan. It's a drug that's derived from the cough syrup Robitussin. It can inhibit these NMDA receptors and drive insulin secretion as well. If you treat cells chronically with these compounds, it leads to them losing their function the next day. But if you were to co-treat with nifedipine, you can protect against the loss of function. So here in the purple bars, this is showing the cell's response to glucose is being protected by nifedipine. And from our 100,000 compound screen, we found a number of other unique chemical structures that are not the same as the SW16789 compound. And this whole class of these compounds cause elevated calcium influx, hypersecretion, and then after an overnight exposure, they cause a loss of function. And in every case, they can be protected. You can protect against this loss of function if you provide nifedipine at the same time. In contrast, the drug thapsigargin, which causes endoplasmic reticulum stress, it acts by a completely different mechanism. It also causes a loss of function after an overnight exposure to beta cells, but nifedipine cannot protect against this loss of function because thapsigargin does not cause stress depending on calcium influx. So we think this hypersecretion stress response could be a unique pathway in the beta cell that's a little bit distinct from canonical ER stress. But when I got to the lab, I still didn't know, when I started my lab, I still didn't know what was the actual protein target of this compound. And so this is the downside of doing these phenotypic screens. You end up with these compounds and you don't know exactly how they work. And so we took a brute force approach to figuring this out. And this was a lot of work done by Geetanjali Roy in my lab. So we wanted to develop a probe that we would be able to use to identify the proteins that interact with this compound. And we had a limited knowledge of different structures that were related to this compound, and we honed in on a region that we thought was amenable to being modified without losing activity. And so that led to the development of this probe called Z629, which we designed with the help of Tim Richardson here at IU. And we used a fluorinated ring here, an aryl azide ring, and the azide is amenable to UV cross-linking. And on the other side of the compound, we attached an alkyne group for click chemistry. And after we made this probe, we confirmed that it had the same activity as the original compound in a chronic treatment. It causes a loss of insulin secretion in response to glucose, which can be protected against if you provide nifedipine. So we felt confident that this probe was exhibiting the same activity as the original compound. Now, the way this target ID experiment works is you put the probe compound on your cells and you expose them to UV light for about 15 minutes. During that time, what happens is the azide turns into a free radical and covalently labels any proteins that are nearby. After that, you can use click chemistry to attach a biotin to this alkyne group, and you have biotin-labeled candidate target proteins that can be purified by streptavidin beads. So we confirmed that in the presence of UV light, we were able to label proteins in beta cells with this probe. And we affinity purified those proteins and submitted them for identification by proteomics. And we performed this experiment not only in mouse beta cells that we are normally using, but we also performed it in the human beta cell line, endo-C beta H1. And because we knew that these cells also responded to our drug. And so this helped us narrow down the potential candidates from this target ID experiment. So we took the overlapping targets that came out of both of these screens. And after some filtering, we narrowed down on this protein called VDAC1. And the reason why this one looked interesting among the other potential targets is that there was already some knowledge about VDAC1 in the literature showing that it's elevated in type 2 diabetes, type 2 diabetic beta cells and could potentially drive some of the beta cell dysfunction. So that appeared to fit with what we are observing with the treatment with our compound too. So we needed to validate that that VDAC1 could be a target of our compound. So we got siRNA and we depleted VDAC1 from beta cells. And we saw that in cells that were depleted of VDAC1, the efficacy of our compound was reduced. So it no longer caused as much hypersecretion as it does in the control cells. And this is sort of in a dose response experiment. Whereas if you knock down VDAC1, the cell's response to just being depolarized wasn't affected. They were still able to secrete insulin in response to depolarization. So calcium influx should be intact in those cells, but it's a response to glucose and the hypersecretion inducer that was reduced. So that wasn't enough, though. We needed some alternative ways to validate this protein as a potential target. So we turned to the thermal shift assay. And so the way this assay works is you incubate your cells with vehicle or your compound, and you put the cells in basically a strip tube in a PCR thermal cycler. And you heat them up to different temperatures, and then you lyse the cells and you do a high-speed spin, and any proteins that are denatured at these higher temperatures will pellet out, and you take the supernatant and you run a Western blot. And if your compound stabilizes the protein of interest, there'll be a right shift in the stability. So it'll basically be soluble at higher temperatures because the compound you're using is stabilizing that protein. And that is what we saw with VDAC1 and SW16789. We saw that the SW16789 was able to right shift the stability of VDAC1 at very high temperatures, suggesting that this is a target of this original compound. And then the next thing to do to confirm these thermal shift assays is to do an isothermal assay under a dose response of your drug. And Gita did that. She took one of the temperatures that looked like we saw a big difference, which was at 64 degrees Celsius, and she incubated cells with or without our compound, and different doses of the compound, heated them all to that temperature, and performed the same assay. And you can see a dose response of increase in VDAC1 stability during treatment with SW16789. So we currently think that SW16789 is at least working through VDAC for part of its activity, and we have preliminary data from one of our collaborators to suggest that SW16789 can cause a slight depolarization of the resting membrane potential that would fit with its ability to potentiate the cell's response to glucose, causing more depolarization, causing more calcium influx. And it was also nice that our collaborators in Michigan and the Les Satin lab were able to confirm some of our results. If they took mouse islets, isolated mouse islets, and measured calcium influx in response to our compound, they could detect a robust response, and they could also detect increased insulin granule exocytosis from mouse islets in response to our compound. So now that we knew sort of the acute, proximal activity of this compound, sort of the targets it might work through and how it's sort of happening to cause calcium influx and hypersecretion, what we still don't understand is how the cells turn off their insulin secretion after a few hours of exposure to this stimuli. So that's what I'll shift gears to and talk about next. So to get at how the cells are responding to this compound over time, we turn to time course RNA-Seq. So we treated cells with vehicle, our SW16789 compound, and we also tested the canonical ER stressor thapsigargan because we're quite interested in how the hypersecretion stress might be different from ER stress. So we treated cells in this time course from 1 to 24 hours, did RNA sequencing, and with all these genes that we discovered a lot of differentially expressed genes, but we needed a way to cluster them to look at their expression over time. So we took two different approaches. In one approach, we collaborated with Mark Keller at University of Wisconsin-Madison to perform WGCNA, which is an unbiased clustering approach. It just takes all the raw data, clusters it into these modules, and then you can look and see which patterns you're observing. And we saw there's this one pattern was a module called Royal Blue that looked pretty specific to our SW16789 compound. And at the same time, we collaborated with Travis Johnson here at IU and IBRI to apply this clustering algorithm called DPGP, which looks at the expression patterns of your genes and clusters them based on these patterns. So in both of these clustering algorithms, we found a similar looking pattern, but the Royal Blue cluster and this SW cluster 3, they didn't have the exact same genes on their list. So we combined these two lists of genes together to generate what we're calling a hypersecretion signature. And if we look at what things were enriched in this signature, one of the top hits is ER stress, of course, but also there were hits for ERAD and a couple other amino acid related peptide pathways. So because ER stress was implicated, we've checked a lot of ER stress readouts. We've done Western blots. We've also done some live cell assays like this one. We used the human beta cell line, endosc beta H1, and we put into the cells a reporter of ER stress that it basically induces a GFP expression when the cells are responding with ER stress. And you can see here the positive control, thapsigargan, for 6 hours. You see here in white, those are cells that are becoming GFP positive. And this is quantified on the right. Thapsigargan will induce a robust ER stress response that's maintained over a 24-hour period. This is going to eventually lead to apoptosis in the cells. Whereas if we treat with our hypersecretion inducing compound, there is a small increase early on, but it quickly subsides and is not very robust of a response. And these studies were in collaboration with the Templin lab here at IBRI. So on top of that, we had checked by Western blot a lot of different readouts of ER stress. I know there are a lot of blots shown here, so let me just direct your attention to the pathways that we did see altered. So in response to hypersecretion, we saw elevated immediate early response genes like CFOS, which are known to be increased in response to calcium influx. So this occurred pretty robustly, whereas thapsigargan did not really induce this response. And we also saw induction of some downstream response genes like the serine metabolism pathway gene PHGDH. However, with thapsigargan-induced ER stress, we can see increased amounts of phospholipid F2 alpha and CHOP and cell death marker like cleave-PARP. These things aren't induced by hypersecretion stress. So, further supporting our idea that hypersecretion is distinct from ER stress, but does have some overlapping features. So because ER stress didn't appear to be largely induced by this treatment, we turned to some of the next pathways that were coming up on our hypersecretion signature, namely the ERAD pathway. So what is ERAD? ERAD is this ER-associated degradation. It's basically like a garbage disposal for the ER lumen. And any proteins that are misfolded in the ER that need to be degraded are tagged by ERAD for export from the endoplasmic reticulum out to the cytosol, and where they are ubiquitinated and degraded. And these proteins that get targeted have to go through a complex that there's a lot of different ERAD complexes, but the major one is this complex made up of cell1L, HRD1, and one of the dirlins, like dirlin 3. And as they're exported, they are ubiquitinated by HRD1, which is the E3 ligase. And in our time course RNA-Seq, we had seen that these ERAD genes were rapidly and robustly induced by SW16789, whereas with ER stress, they are induced, but it takes longer. It takes till 24 hours before those are induced. So we wondered whether this ERAD pathway was responsible for beta cells' ability to adapt to the ER stress and survive and not go through apoptosis. So we took advantage of an inhibitor of this ERAD pathway called iristatin-1. And if we co-treated cells with this iristatin-1 compound and our hypersecretion inducer, we could potentiate the cell death. So normally with either one of these compounds at these doses, there's no increase in caspase activity. But if you combine them, there is a detectable increase in caspase activity, suggesting that ERAD might be helping the cells survive the hypersecretion stress. And on top of that, we also monitored the levels of this ERAD component, and it's also a substrate called OS9. And we found that hypersecretion-inducing compound, SW16789, was able to reduce the abundance of OS9. So this isn't a perfect readout of ERAD. You need to take multiple different approaches to identify alterations in ERAD activity, but it is suggestive that maybe ERAD is being affected by this stress. And there are some other reasons why we thought eRAD could be an interesting player in the hypersecretion response. There are papers from a couple of labs. So this one is from a group at University of Michigan, where they showed that in type 2 diabetic pancreas, cell 1L, one of those components of eRAD, it was reduced in type 2 diabetic beta cells. And another paper from a group in China showed that the E3 ligase, HRD1, was actually induced in diabetes models. So in human islets and in mouse models of diabetes. So it suggests that there's a disconnect in the stoichiometry of these pathway components. In some cases, the components are going down, in some cases, they're going up. So we wanted to take a look at these in our own hands as well. So Gita went into the lab and started staining all of our, we have human pancreas sections from healthy and type 2 diabetic donors. And she stained a lot of different slides and took a lot of different images to measure HRD1 and cell 1L at the same time in these islets. And we used a program called Cell Profiler, which is an open source image analysis program, to measure the ratio between cell 1L and HRD1 only in the beta cells of these samples. And what we could detect is a shift in the ratio. So in type 2 diabetes, it looked like the amount of HRD1 might be going down with respect to cell 1L, which may not entirely agree with what has been found in the literature, but we need to do more work to measure the activity of the eRAD pathway in beta cells. And I mentioned there was other components of eRAD. In one case, there's this component called Durlin-3. It's also induced in hypersecretion, but this one hasn't ever been measured in type 2 diabetic pancreas before. So we stained for Durlin-3 and cell 1L at the same time, and we performed the same kind of analysis so we could look at the ratio of the components. And we found the opposite. In this case, it looked like Durlin-3 was going up, so the ratio of cell 1L Durlin-3 was left shifted. So taken together, what we think is going on is that it could be hypersecretion induced by multiple different drugs, whether it's a sulfonylurea or whether it's the SW16789 compound or other compounds, and whether they act through intermediate targets or directly on membrane depolarization, it doesn't matter in particular, because they all funnel in towards enhancing calcium influx. And once calcium influx is enhanced, you get this hypersecretion response, and there's this feedback that happens, leading to altered gene expression and induction of this hypersecretion signature, as well as a shutdown of beta cell function. And eRAD might be one pathway that is altered in this response and also altered in type 2 diabetes that might help the cells adapt. And if it becomes dysfunctional, maybe it is playing a maladaptive role. So there's still a remaining black box question, which is sort of how does hypersecretion lead to the shutdown of this response? We still didn't figure that out. And it's possible that if we could identify this negative feedback signal, that could represent a new therapeutic target, not only for diabetes, but maybe also for that rare disease I mentioned, congenital hyperinsulinism. So I'll just show some recent preliminary data that we've collected to try to get at this, because this feedback response happens so quickly. It could be due to altered translation. So we do see, because we did see a small change in the ER stress response pathway, but it was transient. So to look at translation, we used this assay called Polyribosomal Fractionation in collaboration with Ron Weck's lab here at IU and his research associate, Jagannath Misra. So what you do in this assay is you treat your cells with your time course and your drugs, and you lyse the cells in sucrose, and you layer them onto this sucrose gradient. You centrifuge them, it fractionates them by size, and mRNAs that are bound to more ribosomes are heavier. They're going to precipitate further down into the gradient. And you use a fractionator hooked up to a UV, an analyzer, and you can then separate out the monosomes and the polysome fractions. And you can see here that worked pretty well in beta cells. And we used dapsogardin treatment as a control. You can see with dapsogardin treatment, the polysome fraction is reduced and the monosomes are increased. That's well known to occur because dapsogardin leads to translation shutdown. And what we noticed with the SW16789 treatment at 1, 2, and 4 hours is that there's a transient decrease in the polysome to monosome ratio, not to the level of dapsogardin, but it is decreased significantly. And then by 4 hours, the cells seem to start recovering. So during this short window, there is likely a difference in translation of certain protein components. And we are working to identify what those might be right now. And just to validate this approach, we showed that some components of ER stress response are actually induced or they're increased their translation even in the face of dapsogardin treatment. And so, for example, here's DDIT3, which is also known as CHOP. That is induced by dapsogardin, and also it's induced by hypersecretion. And you can see here these are increased even when the polysome to monosome ratio is decreased. So with the time I have left here, I will shift gears and tell you a little bit about a totally different project that involves applying machine learning to single cell data in beta cells. And this might inform some of our other experiments on hypersecretion as well. So we started this project in collaboration with Dr. Travis Johnson here at IU. And with the idea that you can learn more about rare subtypes of cells in single cell data if you leverage all the publicly available bulk RNA-Seq data. And so with human islets, there is a lot of bulk RNA-Seq data, and there's also a lot of single cell RNA-Seq data. And what we did, and what Travis did, was he wrote a program called Degas, which stands for Diagnostic Evidence Gauge of Single Cells. And it's a machine learning algorithm that uses deep transfer learning to take information from the bulk RNA-Seq and the donor patient metadata and overlay that onto the single cell map in order to make a prediction about disease association. So we took bulk RNA-Seq data from non-diabetic and type 2 diabetic human islets that was publicly available, along with the donor metadata, and we fed that into his machine learning model. And then we also took a study of 5 different single cell RNA-Seq data sets that have been merged. And we also fed that into the model. And what we were able to do was predict a subset of beta cells that had a high score and some subcells that have a low score for type 2 diabetes association. And this is just some standard slides to show you like how many cells were in this merge, which was graciously provided to us by Mark Housing's lab. There's about 17,000 cells in this single cell human islet merge database. And you can see here the beta cells are labeled by insulin, alpha cells are labeled by glucagon, and there's a good mix of age and BMI across this data set. So I can't really show you what it looks like to run Degas. It's just a computer program that runs sort of in the background, but the output I can show you. And the output looks sort of like this, showing the scores on the cells in like a heat map. So the red scores are a high T2D score, and the black scores are a low score. So you can see here the beta cells have both high and low scoring cells. The low scoring cells might represent the fact that in type 2 diabetes, you actually have a loss of beta cells in general. And so since insulin is one of the major components of beta cells, a loss in beta cells will drive down this score. Nevertheless, we can excise these beta cells from this UMAP plot and plot them by themselves and recluster them. And we can identify the high and the low scoring cells. And in using Seurat, the package used to analyze single cell data, we can compare the high and the low scoring cells and find genes that are enriched in one or the other. And so, after we did that, one of the most enriched genes in the high scoring T2D beta cells is this gene DLK1. And so we used that gene as a candidate to go after first for potentially interesting beta cell subpopulation markers. And DLK1, you can see here, the gene was enriched in the same cells that have the high T2D score. And if you go look into DLK1 on the protein atlas, you can find that it is expressed in human islets, and it does appear to be heterogeneous. There are also publications from like 10 years ago showing that DLK1 also stains in human beta cells and appears to be heterogeneous, but not much has been known about how it might function in type 2 diabetic beta cells. One interesting preprint that came out recently, DLK1 was knocked out in stem cells, and then those stem cells can be differentiated into beta-like cells. And when that was done, the DLK1 knockout cells had increased apoptosis, and they also had a worse response to pro-inflammatory cytokines. So this suggests that DLK1 could provide a protective role in beta cells. And so we wanted to sort of confirm what was seen in the literature as far as normal staining of DLK1 in the islet, but also we wanted to check our type 2 diabetic samples. And so when we stained for DLK1, we confirmed that it is heterogeneous, but to our surprise, in type 2 diabetic samples, it was largely depleted. I'll zoom in here so you can see. You can see here beta cells. There are some beta cells that have DLK1 expression and some that don't, but there are far fewer in the type 2 diabetic samples. And so right now we're working on what's distinct about these DLK1 positive cells. If these cells are resistant to stress, why are they? Are they gone? Them being gone in type 2 diabetes, is that a response to the disease, or is that something else? But we don't actually know all that much just yet about this. And one way we're going to advance these studies in the future is by expanding the application of Degas to a much larger cell set. So when we initially did this, we used the 17,000 cells, but that's really not that many. And now there are way more cell databases available. And so one of Travis's team members, Sylvia Robertson, has merged about 1.3 million human islet cells now. And so we have a much richer database to apply the machine learning algorithm to. And we're in the process of doing that. And once we've run Degas on this, we hope to find probably more hotspots of rare subcell populations within the beta cell cluster that maybe have other rare markers that have gone unnoticed historically because they're in such a small population of cells, but they could mark cells that are going through beta cell failure, or they could mark resilient beta cells and provide new ways to enhance beta cell survival during disease. And with that, I'd like to thank all the human islet donors and the funding organizations from Breakthrough T1D, Congenital Hyperinsulinism International, and my current lab members, Geeta, Andrea, Caitlin, and Rachel, as well as a number of past lab members and all my collaborators who've helped me get to this point. And I'm happy to take any questions.
Fantastic. All right. Thank you so much, Mike. That was awesome. And we have a few questions already. So one of our first questions was, how do these findings translate to the in vivo setting in both mice and humans?
Sure. Thank you. That's a good question. So with SW16789, we haven't tested it in vivo, but there are studies of sulfonylureas that have been done. So in mice, if the studies from Colin Nichols and Maria Rometti at University of Washington, at Washington University in St. Louis, If they have chronic sulfonylureas in an Alzette pump, it eventually causes beta cell dysfunction that's reversible if you take the pump away. And in their recent work, they've found that this could be due to basically a right shifting of the calcium dependence of secretion. And so I think that it possibly fits with the idea that pushing the beta cells to secrete more might not be the best strategy. So we need to find ways to rest beta cells in disease, but obviously we have to do that rest without worsening hyperglycemia. So I think there are opportunities there for future therapeutic development that can sort of suppress beta cell function when it's not needed to give the cells a break, but also allow them to function when glucose levels rise.
Perfect. Okay, and that actually kind of leads on to, I'm going to jump ahead to another question that was linked to that. So if you use this chemical to induce hypersecretion and you gave it to a healthy mouse daily or weekly, would the mouse develop type 1 or type 2 diabetes?
I definitely don't think this would cause type 1 diabetes. That type 1 diabetes is caused by autoimmune attack of the beta cells, and that's by a different mechanism. But to be fair, there are some studies suggesting calcium channel inhibitors like verapamil might be protective of beta cells in type 1 diabetes. So it could be, the beta cell in type 1, if it secretes too much, it could actually be calling attention to itself. It could be producing neoantigens that spur the immune system into attacking it. So suppressing insulin secretion to some extent might be protective of beta cells in type 1 diabetes. When it comes to type 2, I don't think we know. So I think, but in sulfonylurea, in the sulfonylurea induced case, the mice can become glucose intolerant, but I think that's due to beta cell failure induced by the chronic treatment of the drug. So it's not type 2 diabetes classically in the sense that the mice didn't develop insulin resistance. It's just inducing beta cell failure.
Okay, perfect. That makes sense. So what do you think is mediating the negative feedback on insulin secretion?
I think there's some hints in the literature. So from about 20 years ago, there are studies from Jean-Claude Anquin showing that if you give cyclohexamide, which is a protein synthesis inhibitor, even for 2 hours in mouse islets, it leads to a dramatic reduction in insulin secretion. And this happens before the cells lose their insulin content. So they've got the insulin, they've got the granules, but they can't secrete them. And so it suggests that the beta cells are making some protein that either has a short half-life and those proteins haven't been identified, or it's some other nascent protein that just needs to be made quickly on demand to allow the cells to continue responding. And so we hope that using our polyribosomal profiling approach and maybe some other translateomic approaches that we might identify some of these mystery factors.
Perfect. So the next question is a kind of very broad scope question. So in terms of treating chronic diabetes, what's the kind of current standard of care and how is this kind of study modifying that, if you will,
Sure.
or contributing?
I'm not saying that my compound is a therapy. It's more of a tool compound to look at how beta cells function under this stress. It could lead us to new drug targets, though. I see the question. It's sort of, is chronic diabetes treatable? And so I think, patients normally go on metformin and classically maybe on sulfonylureas, but hopefully less and less so. In the future, it seems like the new incretin therapies like Ozempic or Mounjaro, these drugs from Novo and Lilly, are very promising, and they likely can treat a lot of the symptoms of diabetes, because indirectly, if you lose a lot of weight, you're taking the pressure off of the beta cell. So if you reduce insulin resistance, that means the beta cell won't have to work as hard. The earlier you do that, the better. It's probably going to preserve your beta cell function for longer. And then there are also drugs called SGLT2 inhibitors. Those basically allow your kidneys to pee out your glucose. And that's another way to indirectly rest the beta cell, is take away the glucose so that it doesn't have to deal with it. So these kinds of therapies will probably be the best for type 2 diabetes going forward. But there's always going to be people who don't respond to the drugs, or they can't afford the drugs. And so that leaves a lot of space open for us to develop novel therapies.
Absolutely. Great. So the next question is, have you tried single-cell RNA-seq on DLK1 knockout samples, and what are the downstream transcriptomic effects?
Yeah, that's an expensive experiment. So I don't have the DLK1 knockout animals. And the group that made the knockout stem cells, to my knowledge, I don't know if they've done single-cell RNA-seq on those samples. That's Shui-Bing Chen's group and Francis Collins's group. And what you would expect, actually, what I'll be able to get at that, actually. So what we're going to do in one of our future projects is we're going to try to separate from primary human islets. We're going to try to separate the DLK1 positive and DLK1 negative cells, and then we can do RNA-seq on those. I mean, another thing you can do is you can just look at the existing RNA-seq, the existing single-cell RNA-seq, and find the DLK1 positive cells, which is sort of like the population of high-scoring diabetes cells we identified. And you can just look at the genes that are enriched in the same cell that correlate with DLK1 expression. The downside of single cell is that the read depth is only giving you about 5,000 of the top expressed genes. You might be missing lower expressed genes like GPCRs, for example. So you might not pick up a lot of the other 10,000 genes that single cell the genes that are expressed too low a level for single cell to pick up. So in those cases, you might need to do like a purification of the cells and do a bulk RNA-seq on the purified cells. I see there's one other question. I think I lost Connie, but I see one other question. Someone asked if Degas is more effective than other algorithms in detecting rare expressed genes in single-cell RNA-seq. I mean, we think so. Travis has done a good job characterizing his application. He's used Degas in Alzheimer's. He's used it in cancer samples, and they've been able to identify rare subtypes of cells. Now, there are other algorithms. So his algorithm is a nonlinear model, but there are other like linear model-based algorithms. There's one called Scissor, and there's some other ones that basically try to do some of the same things. I think taking a combinatorial approach, running all of the different algorithms is a good idea. And see if you see the same cell type the same subset of cells being highlighted by more than one of your algorithms, I think that suggests that it's a real subtype. Samhita, do you know if we lost Connie?
Yeah, we lost Connie. Her internet crashed. Yeah.
That's okay. I think we actually answered all of the questions.
Yes. If there are any last minute questions, feel free to send them, but I think we've answered all of it. Awesome. Thank you so much. That was a really wonderful presentation. And thanks everybody for coming out and listening to this talk.
Yeah, thank you very much.